Growth hormone, commonly referred to as GH, has become a significant topic of interest due to its complex role in the body, especially concerning exercise and resistance training. GH is far more intricate than initially understood. For many years, most research on GH focused on the 22 kilodalton form of the hormone, 191 amino acid polypeptide produced in the somatotrophs of the anterior pituitary gland. This specific form, called monomer, has been the most studied because it can be easily measured using radioimmunoassays. However, recent discoveries show that GH exists in various forms, including aggregates where multiple GH monomers are linked together through disulfide bonds. These aggregates can be found in the blood in much higher concentrations than the 22 kilodalton form, and they may have even greater biological significance. The focus on the 22 kilodalton form for so long limited our understanding of GH's full impact. Until recently, researchers missed much of what was happening with these larger GH aggregates. It has become apparent that these aggregated forms may play critical roles in the body's adaptation to exercise. There is even evidence to suggest that the aggregate forms of GH might be the primary biologically functional variants responsible for these adaptations, rather than the 22 kilodalton monomer. Most studies in exercise endocrinology have continued to focus on the 22 kilodalton form of GH due to the ease of measuring it through common immunoassay techniques such as radioimmunoassay, RIA, and enzyme-linked immunosorbent assay, ELISA. These techniques rely on antibodies that are generally specific to the 22 kilodalton variant, which means other forms of GH, such as aggregates, have often gone undetected. The importance of GH and its many forms is underscored by its wide range of physiological roles, which include decreasing glucose utilization, increasing protein synthesis, breaking down fats, and enhancing immune cell function. While GH is crucial for normal development in children, it also plays an essential role in how adults respond to the stresses of resistance training. For example, a study by McCall and colleagues found a positive correlation between GH responses and muscle fiber hypertrophy following 20 weeks of resistance training. Although this study only looked at the 22 kilodalton form of GH, it is likely that other forms of GH also contributed to muscle growth and adaptation. GH's effects are widespread, with target tissues that include bone, skeletal muscle, immune cells, fat cells, and the liver. Among its key roles are increasing protein synthesis, promoting collagen synthesis, and stimulating cartilage growth. The secretion of GH is regulated by a complex system of neuroendocrine feedback mechanisms. The 22 kilodalton form stimulates the release of insulin-like growth factor 1, IGF-1, at the autocrine level, which plays a key role in muscle protein synthesis and tissue repair, particularly following resistance exercise. IGF-1 may be released from various tissues in the body, including muscle, fat, and white blood cells, although muscles themselves typically do not produce as much IGF-1 as other tissues. GH secretion follows a pulsatile pattern with bursts occurring throughout the day. The highest levels of GH are usually observed at night during sleep, and exercise can increase both the frequency and amplitude of these bursts. This pulsatile release mainly applies to the 22 kilodalton form of GH, whereas bioactive GH aggregates do not seem to follow a circadian rhythm. It is believed that the nocturnal increases in GH are tied to tissue repair processes, especially after the stress of exercise. Pituitary hormones like GH respond to a variety of exercise stressors, including resistance training. It has been shown that even breath holding, hyperventilation, and hypoxia can increase 22 kilodalton GH concentrations. However, an intensity threshold must be reached for significant GH responses to occur during resistance exercise, especially when longer rest periods are used. So studies have demonstrated that short rest periods between sets combined with higher training volumes produce large increases in 22 kilodalton GH concentrations. For example, Kramer and colleagues found that performing 3 sets of exercises at a 10 repetition maximum and 10 RM with short 1-minute rest periods resulted in substantial increases in GH levels, particularly when compared to longer rest periods of 3 minutes. Growth hormone responses also differ between men and women. Women generally have higher baseline concentrations of the 22 kilodalton form due to greater frequency and amplitude of GH pulses. Additionally, hormone responses to exercise in women can vary depending on the phase of the menstrual cycle. Research by Kramer and colleagues showed that women had significantly higher resting 22 kilodalton GH levels during the early follicular phase of the menstrual cycle compared to men. Interestingly, despite these higher baseline levels, both men and women experience similar increases in GH following resistance exercise, provided the exercise protocols are designed with short rest periods and moderate resistance. For women, resistance training has been shown to increase concentrations of bioactive GH. However, this effect seems to diminish with age, as older women tend to have lower concentrations of bioactive GH than younger women. Strength-trained women generally have higher bioactive GH levels than sedentary women, suggesting that the aggregated forms of GH play a crucial role in muscle adaptation and strength development. The complexity of GH, especially the larger bioactive forms, underscores the need for further research in this area. The adaptive responses of GH to resistance training have not been extensively studied, but it appears that training reduces the GH response to exercise over time. In elite weightlifters, long-term training has been shown to reduce the exercise-induced GH response, while resting levels of bioactive GH remain relatively stable. This suggests that other GH forms, possibly the larger aggregates, become more important as athletes progress in their training. Insulin-like growth factor 1, IGF-1, is another key hormone closely tied to the actions of GH. IGF-1, a 70 amino acid polypeptide, is produced primarily in response to GH stimulation, particularly from the liver, but also from other tissues like fat and muscle. Like GH, IGF-1 plays a crucial role in muscle growth and repair by promoting protein synthesis. IGF-1 travels in the blood bound to various binding proteins, which regulate its availability for interacting with receptors in target tissues. There are at least 6 different types of IGF-1 binding proteins, and each one responds to exercise stress in different ways. These binding proteins act as transporters, restricting IGF-1's access to receptors until it is needed. IGF-1 can be produced within muscle cells in response to mechanical stimulation, like stretching or overload during resistance training. This local production of IGF-1, often referred to as growth factor, is believed to be the main mechanism by which muscles adapt and grow in response to resistance training. Acute increases in IGF-1 following resistance exercise are likely tied to the disruption of muscle and fat cells, both of which contain IGF-1. The exact reasons behind these increases remain unclear, but it has been suggested that mechanical stress and cellular damage play significant roles. IGF-1 responses to exercise can vary depending on baseline concentrations. If baseline IGF-1 levels are low, exercise tends to increase its concentration. However, when baseline levels are high, exercise may not cause a significant rise in IGF-1. This variability has been demonstrated in studies where resistance exercise led to increases in IGF-1 in participants with lower starting levels, while those with higher baseline IGF-1 concentrations showed little or no change. Nutritional factors also influence IGF-1 responses to exercise. Consuming protein and carbohydrates before and after exercise can enhance IGF-1 responses, as demonstrated in a study by Kramer and colleagues. This suggests that caloric intake plays a crucial role in regulating IGF-1 levels, especially in the context of resistance training. Overall, both growth hormone and IGF-1 are vital to the body's adaptation to resistance training. GH exists in various forms, each playing a unique role in promoting tissue repair, muscle growth, and fat metabolism. Similarly, IGF-1 is critical for protein synthesis and muscle adaptation, operating through complex autocrine and paracrine mechanisms. As research progresses, our understanding of these hormones' roles in exercise adaptation will continue to evolve, revealing new insights into how the body responds to physical stress and training.